Okay.
We are recording this seminar just so everybody's aware.
Good afternoon, everyone. Thank you for letting me come and talk to you this afternoon. For you, it's the morning for us still. What I'm going to do today is firstly take you through a little bit of cancer biology, so a background to tumour cell biology, and then focus in on the aspects of infectious cancers with a particular emphasis on lymphoid cancer, so cancer of the immune system. So, as I'm sure you're all aware, cancer isn't just one disease, it's hundreds if not thousands of different diseases because we're all very genetically diverse. And in the UK, you can see that the majority of cancers that we see in the clinic are breast cancers, and most of those are in women, followed by lung, colorectal and prostate cancer. So they're the big four cancers that we see over here. And as we move down the list, you'll see that the sixth most common cancer there is something called NHL, or non-Hodgkin lymphoma, which is a cancer of the immune system. And that's what my research focuses on over here in Cambridge. And so I'm just going to briefly take you through what cancer is. But before I do that, I'm also going to tell you a little bit about childhood cancer, because that's what my research focuses on. And here you can see the incidences of childhood cancer across the world. And North Africa is up there on the top, followed by sub-Saharan Africa, and the incidences of childhood cancer. And as we move down, you see Europe there. And what you can see instantly is that children don't get the same types of cancers that adults get. So children mostly get leukemias, which is a cancer of the blood, lymphoma, a cancer of the immune system, central nervous system tumours, so brain cancers, and then some other rarer cancers, which are shown in the slide on the right there. And so these are things that are generally cancers associated with problems with development. So things such as retinoblastoma, which is a tumour of the retina, renal tumours such as Wilms tumour, so cancers affecting the kidneys, germ cell tumours, so really developmental origin tumours. So different range of tumours in children to what we see in adults. And I actually managed to find some data on cancer incidences in Kampala as well. So you'll see in Kampala, you have a slightly different range of cancers than we see over here in the UK. And we'll get on to why that might be the case shortly. But you can see here instantly that the most common cancer in women is cervical cancer, followed closely by breast cancer. And in men, the most common cancer is prostate cancer, which is very similar to here in the Western world, but that's followed by Kaposi sarcoma and esophageal cancer. So a different range of tumours. And if we look at why that might be, what we can see instantly is most of the types of cancers you see over in Kampala are associated with infections. So cervical cancer is associated with HPV infection. Bladder cancer with schistosomiasis, hematobium infection, nasopharynx, cancer of the nasopharynx associated with Epstein-Barr virus, as are many of the non-Hodgkin's lymphomas. So for non-Hodgkin's lymphoma, we see them often in patients in Kampala with Epstein-Barr virus, HIV, HTLV, and a particular bacteria called Helicobacter pylori. And I'm going to talk about that in more detail later. Liver cancers, of course, are associated with hepatitis infection, so hepatitis B and hepatitis C virus. Esophageal cancer, again, with HPV. And of course, Kaposi sarcoma, which you'll know to be more commonly seen in patients with HIV or Kaposi sarcoma herpes virus. So a lot of virally or bacterially or parasite-induced cancers are more often seen in Uganda than we see over in the Western world. And this doesn't change much for children. So children in Kampala, a large majority of children are diagnosed with lymphoma, cancer of the immune system. And about half of those children who have lymphoma in Kampala will have Burkitt lymphoma, which some of you may be familiar with. It's this large tumour that we see in the jaw of children and is associated with EBV infection. So what is cancer? We know it can occur in many different body types, organs within the body. And what we know is it's an abnormal growth of cells. So essentially you get a tumour, a mass of cells, which has essentially lost control. So the cells within that tumour no longer have the growth controls which stop them from growing out of control. And in that regard, they can become invasive. And invasive growth is the definition of what is a malignancy, a tumour. So a malignancy, a cancer, is when the cells become invasive and can invade into other parts of the body. In some cases, they can also become metastatic. So that is, they're able to move through the lymphatic system or through the blood to other sites within the body and set up new tumours, which we call metastases. And ultimately, this loss of growth control is the consequence of a combination of genetic mutations. So at its heart, cancer is a genetic disease. And how those genetic mutations are acquired, we'll go into shortly. So just to put that into perspective and to think about growth control, if we just think about our skin, we think of it as a layer of cells that's protecting the tissue underneath. But if we look at the cartoon, you can see that that tissue is a bit more complex than just a layer of cells. It actually consists of a number of different parts of the body. So we have blood vessels, which are the blue and the red lines there, which we see moving up towards the tissue spaces. We have sweat glands, so you can see that little white knotty shape there down at the bottom end is a sweat gland. And then you have the layers of skin. Of course, skin is made of keratinocytes, and at the base level there, at the base of the cells, those cells which are highlighted in that brown colour are the stem cells. And the stem cells are the only cells that have the ability to self-renew and also to give rise to progeny. And of course, as they produce those progeny, those cells are what form the skin. And as they mature and differentiate, they move up towards the surface of the skin, and then they get sloughed away as they die. And of course, this is the way our body protects ourselves. This is our most innate barrier to infection, our skin. And only through constantly replacing those cells as they die do we maintain that barrier. Now, if that growth control is lost, so for example, if we don't produce enough skin cells, we will have problems, we'll have breaches in the barrier. And if we produce too many skin cells, again, that can cause problems. But if we produce too many skin cells and those cells don't mature and differentiate and die, then we are at risk of developing a malignancy, a cancer. And on the third image across from the left there, you'll see this is a microscopic image of skin. And again, you can see those different layers of skin cells. They start off with the stem cells at the bottom, at the basal layer. They differentiate, and as they differentiate and mature, they die, and they're dead at the surface there and get sloughed away. So what's controlling all that growth? What's controlling all the growth properties and telling those cells to die when they get to the top, and the cells at the bottom to divide and replenish themselves, and also to give rise to new cells? What's telling them to do that? And of course, we know it's those cells within each of those cells is DNA, and that DNA code is what is informing, instructing the cells as to how they should behave. And so if there are mutations in the DNA and there are mutations in genes which elicit that growth control, you can imagine that there can then be problems and a risk of malignancy. So to put it in a very simplistic format, our cells all contain a DNA code. That DNA code is duplicated into the new cells, the daughter cells, as they divide and develop. And that process goes on as those cells develop further and divide more. But if we should have mutations in that DNA, those mutations will then be propagated down further into the daughter cells. So what causes those DNA faults and that damage? Well, we know that there are natural cell processes which give rise to DNA damage. So we know, for example, just through metabolism, our cells produce reactive oxygen species. And we know that reactive oxygen species can induce DNA damage. We also know that just in that process of replicating your DNA to share it between daughter cells, that there can be DNA error. There can be error in those DNA pathways. But what we also know is that's normally repaired because we have multiple DNA repair pathways to prevent mutations from propagating further. So there are natural cell processes, but we also know that other things can affect the rate of DNA mutations. So sometimes in our environments, we're exposed to mutagens or carcinogens. Perhaps we work with pesticides, or we work with petrochemicals, or work with other things which we know can induce DNA damage. Maybe in our lifestyle, we do things which we know can induce DNA damage. So particularly for people with fair skin, exposed to the sunlight, there's a higher risk of inducing thymine dimers in our DNA. And we know that these are mutagenic. And if they happen to occur in genes that play important functions in cell control, they can therefore predispose to malignancy. So there are environmental factors that can also induce mutations. We know that viruses can also induce mutations, and in particular, I'm talking there about viruses that integrate their genome into our genome, so retroviruses in particular. And if they integrate their genome into areas of our DNA that encode important genes in growth control, again, that could be mutagenic. At the bottom there is genetic inheritance. Genetic inheritance of cancer is exceptionally rare, but it does happen. For example, we know that some families already have mutations in genes involved in DNA damage repair, such as BRCA, the BRCA genes in breast cancer, or have mutations in genes that control cell growth, such as P53. And these are our so-called Li-Fraumeni families. And if you follow through the family trees, you will find that many of the members of the family develop cancer. But that's exceptionally rare. So as I said, we have this growth control. And so normally when DNA damage is sensed by machinery in the cell, so by the proteins such as ATM, ATR, which detect double or single strand DNA breaks, this triggers a process whereby cell cycle arrest occurs. So proteins such as P53 or retinoblastoma, RB, stop cell division and allow the cell to pause to either repair that DNA damage, or if the damage is extensive, to trigger an apoptotic pathway. And so that's depicted there by the pictures on the left. P53 will trigger cell cycle arrest. The cells will either then repair their DNA damage, or if it's too much, they'll undergo apoptosis. And this is to prevent the propagation of those mutations down through further generations of cells. Now, if there should be a failure in those processes, of course, those mutations are then going to be propagated through further cells, and those cells can in time then pick up even more mutations, which could be in genes that are associated with growth control, and therefore again predisposed to malignancy. So when we think about cancer, we're talking about a series of gene changes, changes in the gene. They may either be mutations at the level of the single amino acids, they may be large mutations as a consequence of changing of genetic materials such as chromosomal translocations, inversions, insertions. They may even be at the epigenetic level. So for example, changes in CpG methylation within promoter elements, which determine whether or not a gene is expressed or not. And as a cell acquires more and more of these mutations, and if these mutations are in genes which are important for controlling cell growth, that cell may become more and more abnormal and eventually acquire the ability to self-renew, much like a stem cell, to proliferate out of control, to switch off those pathways which determine whether a cell will live or die, and to grow out of control and develop into a malignancy. Okay, so what genes are we talking about here? So some of those mutations may occur in genes of which there is no consequence, because perhaps those genes aren't used by that cell. So if there's a mutation, we don't care. The cell doesn't care because it's not expressing that gene, so it's of no consequence. But sometimes those mutations may occur in genes which, as I said, confer growth control. And we call these oncogenes and tumor suppressor genes. So oncogenes are proteins whereby they get activated and switched on due to that mutational event, and that gives them a gain of function. So they gain some sort of activity through that mutation. Normally, these are genes which are associated with proliferation. So if these cells get switched on, these mutations switch on these proteins so that they become constitutively active, this obviously has consequences for the cell. And mutations in oncogenes normally affect just one allele. So of course, every gene comes as 2 copies. and they have a dominant effect. So this gene becomes switched on, it becomes constitutively hyperactive protein, which is constantly driving cell proliferation. On the other end of the spectrum, we can also have mutations which disable tumor suppressor genes. So tumor suppressor genes are those genes which normally act to constrain growth control. And so in this case, the mutations that occur lead to loss of function. So we lose the function of those genes so they can no longer constrain growth and proliferation. And in this case, we normally have to have the mutation in both copies of the gene, because often if you just mutate one copy of the gene, that second gene still produces the normal wild type protein, and in many cases it can carry out the normal function. So often you need to lose both copies in the case of a tumor suppressor gene. So they're recessive in the cell. And these are things such as P53, which induces a cell cycle arrest, and also retinoblastoma. So if we look at the retinoblastoma protein here, retinoblastoma is a protein which initiates the G1S checkpoint. So if retinoblastoma is not phosphorylated, so in its unphosphorylated format, it is able to induce a G1S cell cycle arrest. And of course, there's a pathway of proteins which link into retinoblastoma and into the cell cycle. And it all starts upstream there with P16. P16 is also a tumor suppressor. So if that gets mutated in any way, it can no longer initiate this cell cycle arrest. On the other hand, the two proteins in orange there, the cell-dependent kinases, cyclin-dependent kinases and cyclins are oncogenes, because these have the effect of phosphorylating and inhibiting tumor suppressor genes. So if you're unable to do that again, you can switch on this hyperactive proliferative pathway whereby RB is not being engaged to arrest the cell cycle. So we can have activation of oncogenes, deactivation of tumor suppressor genes, anyway within a cell growth pathway. And as long as that all has the same downstream effect, which is to prevent cell cycle arrest, this can be a tumor promoting activity. So what happens is those mutations they get accrued in cells so that those cells move along the pathway to becoming more and more abnormal and to losing growth control. And so there is a particular model of colorectal cancer, and this is the model that was put forward by Bert Vogelstein many years ago now, whereby he could look at the genetic mutations in precancerous growths all the way through to a cancer and to its metastasis. And he could find that within normal cells, which have relatively few mutations, certainly no tumor promoting mutations, that the first step towards developing a small adenoma, and an adenoma is essentially a polyp within your colon, and that polyp is a precancerous growth because it has a mutation in APC or beta-catenin. And these are tumor suppressor genes, and so those mutations already predispose these cells to losing growth control and becoming more and more malignant. And he could link this series of events whereby the cells would then lose CDC4, they would then gain activity of oncogenes such as KRAS or BRAF, then gain activity of further oncogenes such as PI3 kinase or lose the tumor suppressor P10, gain mutations in tumor suppressors such as P53 and SMAD4, and eventually that would be enough mutations to enable the cells to completely lose growth control and develop into a malignancy. Now, what then causes metastases to occur is a whole other matter, which I won't go into today. But essentially, the cells acquire the ability to become tumor cells through gaining the hallmarks of cancer. And the hallmarks of cancer were proposed by Hanahan and Weinberg many years ago again. And this is work that was published initially in 2000, where they proposed that cells have to acquire enough mutations in pathways that control multiple aspects of cell growth. So the cells have to acquire mutations that give them the ability to evade apoptosis. They have to be self-sufficient in growth signals. They have to become insensitive to anti-growth signals. In some cases, they have to acquire the ability to metastasize, but certainly the ability to invade into tissue spaces. They have to acquire the ability to have limitless replicative potential. And as this tumour grows and expands, it needs more and more food and nutrients. So therefore, it must also acquire the ability for sustained angiogenesis. So the growth of blood vessels into the tumour, the growing tumour, to provide food and nutrients for this growing mass. And how those cells acquire those particular pathways doesn't necessarily need to be in any particular order, but those pathways are required through mutations. So for example, self-sufficiency in growth signals could be acquired through an activating mutation of the RAS oncogene. So RAS requires ATP to provide energy to activate the MAP kinase pathway. And so any mutation which locks in ATP in that binding pocket and causes it to be hyperactive would give that cell the ability to be self-sufficient in growth. It doesn't need any messages to tell it to grow anymore, doesn't need ligand to bind to the cell, because that RAS-MAP kinase pathway is active all the time. It could lose sensitivity to anti-growth signals, so signals that would tell the cell to stop dividing by losing that retinoblastoma tumor suppressor. Another example to evade apoptosis would be to produce its own survival factors. So instead of relying on growth factors coming in from outside the cell, it can switch on pathways where it produces its own growth factors, which then are secreted, bind to receptors on the cell surface, and it can do this in an autonomous manner. Limitless replicative potential. So one thing it can do there is turn on telomerase, so an activating mutation in telomerase, so that it maintains its telomeres, the telomeres remain long, and therefore it has this limitless replicative potential. Angiogenesis. How would a cell sustain angiogenesis? Well, one thing it might do is produce vascular endothelial growth factor, or VEGF, perhaps through a mutation somewhere in that pathway or within VEGF itself, so that again, it produces its own vascular endothelial growth factor. This gets secreted from the cells and recruits the growth of blood vessels to the tumour. What about tumour invasion? Well, if you're an epithelial-derived cell, you may inactivate E-cadherin. So E-cadherin forms those links between cells, and if you lose those links, you're no longer tied to your neighbour cell anymore, and you can then invade and move into other tissue spaces. So we need genetic mutations. We need them in certain genes, in certain pathways, which give us these properties to become a malignancy. Now, as well as these properties, there have been some further enabling properties that have been published in an update to the hallmarks of cancer. And these are perhaps 5 properties which rather than are required for malignancy, perhaps enable it to progress further. And these properties are the ability to evade the immune system. So we have an immune system, and we have things such as NK cells, as you will know, and we have T cells, which are constantly surveying the body and are on the lookout for abnormal growth of our own cells, as well as looking for invading pathogens. And so a cell has to acquire the ability to evade that activity to become a tumour. We know that they have to be able to cope with DNA damage and DNA replication stress, because if you're a rapidly dividing cell, you're rapidly replicating your DNA, and you are more prone to develop DNA damage, which can be a good thing, because then you'll get more mutations in more genes, and that could help you to grow further. But also, if you have too much DNA damage, that could lead to cell death, unless you have activation of those anti-apoptotic pathways. So there are these properties as well that are required. And of course, if you're a rapidly dividing cell, you're also going to be exposed to more oxidative stress, mitotic stress, proteotoxic stress, and metabolic stress. So these cells have to acquire the ability to cope with all these stresses. So we have multiple pathways to cancer. We have the enabling properties. We have the hallmarks that need to be acquired through genetic mutations. But what determines in which cell types this occurs and which cell types are more predisposed to cancer development? So you can imagine cell types that already rapidly divide, such as the lining of your intestines, such as your blood cells, they're also rapidly dividing, are perhaps already more prone to DNA damage, and therefore maybe that's why we see cancers, particularly in those cell types. But for most cancers, we don't know what the cell type was where the initiating event occurs. So we assume that amongst cancers, there is one genetic mutation which initiates this pathway, this snowballing of accumulation of genetic events. But we don't know where it occurs. We don't know at what stage development it occurs. We don't often know what causes it. And for pediatric cancers, we often think that maybe for some pediatric cancers, those initiating events actually occur in utero in the pregnant mother. So this cell type, which we don't know about, and in the case of lymphoma, a cancer of the immune system, presumably the cell it all starts in is a lymphocyte. And we know that lymphocytes, in the picture there, you can see on the top left, are in our blood, they're in our lymphatics, they're circulating there amongst red blood cells, and they're there to survey our bodies and look for invading pathogens and look for abnormal growth. But presumably, some sort of genetic event occurs in that cell, which predisposes it to acquiring further mutations, which would give rise to those hallmarks of cancer and lead to an established growth that propagates. And on the bottom left there, you can see a little boy who has large tumor growth in his neck, in the lymph nodes that are in the neck. And generally, we all get swollen glands in our neck, in our groin, in our armpits. And that's normally a response to infection. And normally that will go down, that swelling will go down within a couple of weeks as the immune cells die off as they've responded and cleared the infection. Of course, in the case of lymphoma, that doesn't happen. So in the case of lymphoma, when those swellings stay around for a month, 2 months, then it's time to go to the doctor and try and work out whether this is just a residual inflammatory response or if it is something more sinister, such as a lymphoma. Okay, of course, what we have to remember in all this is cancer cells have not been designed, they have evolved. So very much like we think about evolution, cancer cells also go through a mini process of evolution within themselves. And those cells which have mutations which give them enabling properties are the ones that are going to survive and develop. And so they're not necessarily going to have an ordered process of genetic events. They're just going to be the ones that survive. The fittest clones that develop these mutations are going to be the ones that evolve and develop into malignancies. So I just want to bring to your attention a couple of papers that were written by Vogelstein in 2015 and 2017, where they had done a lot of genetic studies of lots of different types of cancers and could put cancers into 2 different categories. So one they called a set of cancers that developed due to bad luck, and one a set of cancers that they said were due to environmental and inherited factors. And so you can see the ones that are due to environmental and inherited factors are those associated with, for example, viral infection, or associated with exposure to radiation, or lung cancer and smoking, or cancers associated with a genetic predisposition, such as some colorectal cancers. And then on the ones on the left, they said we're down to bad luck in that you just happen to acquire mutations in the genes that control cell growth in the right cell type at the wrong time, and therefore just get tumor growth. And this was the idea that if you think about we've all heard of stories of people who've smoked 60 cigarettes every day for their whole life and never developed lung cancer. So this was their way of trying to account for those people, that maybe the cancers that aren't being developed in those people that smoke is because they're not getting mutations in the growth control genes for whatever reason. So this led to a lot of press interest. And the press interest was along the lines of, oh, okay, if it's just down to bad luck, that means I can smoke. And I can smoke in front of my children and you know, I may just get lucky and not develop cancer. It led to other things, such as people saying, well, my granddad's nearly 100. He smoked for over 30 years. He gave it up, but he's well. He walks very fast. He does Tai Chi, and he eats garlic and ginger. So therefore, if you do all those things, you're not going to get cancer. Is that the case? And there's a report that came out in the Daily Mail, which is a newspaper in the UK, and they read this paper and said, well, most cancers are caused by bad luck, not lifestyle. So let's just live our lives and not worry about cancer. But that's not really the case. That's not really what the paper was saying. Because we all know we get mutations in our DNA all the time through natural processes. Many of those will be repaired. But we also know that we can expose ourselves to things in the environment which will increase our chances of getting mutations in those growth control genes. So essentially, you want to control your risks. And the way to control your risk is to not do those things which we know cause mutations. So not to smoke, to eat the correct diet, to not expose ourselves to the sun. We know some of those things are out of control. We know some cancers are hormonally induced, such as breast cancers and ovarian cancers. We know some cancers are due to infections, and it may be that we can't control whether or not we get infected, but there are now vaccinations, as you will know, against some things such as hepatitis viruses. So infections could prevent the risk of liver cancer. So there is a lot we can do in that roulette wheel of cancer to move our risk of developing cancer back down to the lower end, rather than doing things which moves us up to that higher end, which increase our chances of cancer development. So it's a genetic disease. It starts with cells which lose growth control, which start to take up space within our body. And when at these very early stages, it can be hard to detect cancers because they have no effect on the rest of the body, so we have no symptoms. But as they get bigger and more aggressive, they start to do different things. So as I said, when oxygen becomes a problem, when food becomes a problem, they secrete those growth factors such as vascular endothelial growth factor to tell blood vessels to move and grow up towards them. And so you can see that there in the third image, that the cells have essentially tricked the blood system into thinking that they are a new growth, that they are essentially cells repairing damage perhaps, and to provide them with the nutrients they need to keep growing bigger and bigger. And of course, as this process continues, as well as, if we look in the bottom picture there, as well as cells being able to move up into the blood, into the tumor, and provide nutrients and oxygen, tumor cells can equally move down into the blood supply and hence move to other parts of the body and seed metastases. So this is generally when we start to diagnose cancers, is when they're at these latter stages here, when they're much more aggressive, but now they're starting to cause symptoms because they're getting bigger and affecting the function of the cells around them. Okay, so we like to think of a malignancy as just a growth of cells, but we know it's not just that. And that's why a lot of treatments fail, is because we're just thinking about killing those cancer cells. But there are other aspects of cancer cells that we need to think about. And within a tumor, we don't just have cancer cells, we have angiogenesis, so we have that blood supply. So another way we might think about treating cancers is to cut off that blood supply. We also know that there are immune cells which infiltrate into tumors. And those immune cells actually, in many cases, provide growth factors to help the tumor to grow. And these will be cells like T cells, and NK cells, for example. There will also be B cells there, but these cells can actually play a different role to what we would hope they would do and actually enhance further cell growth. And often that's also because, of course, the cancer cells evade the immune system by upregulating expression of proteins such as PD-1. We also know that there's a growth matrix there within the tumor. So often there'll also be fibroblasts, and fibroblasts are producing a stroma that provides a sort of meshwork, a basket work for those tumor cells to grow within. So there are a lot of different things we need to think about when we think about a tumor. And I just want to show you what one looks like. So on the left here, we've got a normal healthy colon. And you can see it's shiny on the inside there, and it's shiny because there are glands around it producing mucus. And the role of that mucus is to help stools, feces, move through the colon. Now on the outside is muscle, and there is muscle in two directions, and that undergoes something called peristalsis. And the reason for that is that muscle then pushes the stools through, aided by the mucus, and evacuates the stools. And you can see in the microscope image on the bottom there, the glands are down the bottom, and look and see how nicely ordered they are. They're all round, or they're longitudinal in shape, and they're producing mucus and secreting it into the colon. And you can see the light pink muscle layer there. It's nice and continuous. You've got your longitudinal muscle and your circular muscle. They're at the top there. And in the middle is a stromal layer, a layer of connective tissue that just keeps it all together. Now, if we move to the image on the right, you can instantly see there's a growth. There's a mass of cells in the lumen of the colon. And it's a pretty nasty, angry looking growth. There's lots of mucus there. And you can imagine that this is the stage at which we start to diagnose colon cancers, because if you have stools, feces, trying to push past that growth in the colon, they're going to tear it, and therefore you'll get blood in the stools. And often that's the first sign of colorectal cancer, is blood in the stools, and also on further examination, some of the tumor cells which get sloughed off as the stools push through. But it's a nasty growth. Now, how do I know it's a cancer? If I look at that microscope image of the tumor down on the bottom there, and I'm going to enlarge it for you here, and the area on the right is the relatively normal part. So that's the nice ordered glands, the nice circular longitudinal shapes producing the mucus. You've got the nice pink muscle at the top. You've got that nice layer of connective tissue in between. Really well ordered. There's order to the tissue. Now, as we move to the left, you can instantly see you've lost that order, that structure. And in particular, you've lost that nice layer of stroma that separates the two. And that's because the glandular tissue at the bottom left there is very disarrayed. You can see it's not arranged in nice little circles or oblongs anymore. It's now all sorts of anaplastic shapes. So anaplastic just means abnormal looking. And we've lost that nice layer of stroma because it's invaded into the underlying muscle, that nice pink muscle. So this is now a malignancy. It's no longer benign. It is a growth of cells which is invading into the muscle. And that invasion is important in the pathology of cancer because when it invades up into the muscle, it can move up to the blood vessels as well, and it can get into the blood vessels. And we know that the colon is attached to the liver through the hepatic portal vein. And of course, if it can get into the liver, it can set up a metastasis. So this pale pink lump in the middle, that's glandular tissue, and you shouldn't have glands in the liver. So we know that it's a metastasis, and it most likely moved through the portal vein from the colon into the liver. Okay, so hopefully that's given you a reasonable background to cancer biology and an understanding of why tumours develop in the first place. And now I'm going to move into lymphoma in particular and cancers of the immune system. Now, cancers of the immune system develop from the white blood cells, not from the red blood cells, because as we know, red blood cells have no DNA, because red blood cells live for a very short period of time and then they die. And so we never see cancers of red blood cells. What we see cancers of are white blood cells. So NK cells, we see cancers of B cells, T cells, and also myeloid cells. Now, cancers of myeloid cells are always leukemias, and they circulate in the blood, and that's a liquid tumor. Cancers of lymphoid cells, such as B cells and T cells, can either be lymphomas or leukemias. If they're leukemias, they're a liquid tumor. If they're lymphomas, they're a solid tumor. So we're dealing with essentially all those cell types. When we look at that hierarchy from the bone marrow stem cell, we're dealing with both the myeloid and the lymphoid arms when we think about leukemias and lymphomas. And I'm going to focus on the lymphoid arm and the lymphomas because that's where my particular research interest is. So again, much like I've just talked about in all the other cancers, these cells acquire mutations within their DNA which can predispose somebody to developing a leukemia or a lymphoma. I just want to show you what a leukemia looks like. So on the left is a normal blood smear. So you can see most of the blood is made up of red blood cells. And the odd blue cell there, they're lymphocytes. They could be B cells, they could be T cells. We wouldn't know unless we stain for, say, CD4 for a T cell or CD20 for a B cell. But there are lymphoid cells there. And in the leukemia on the right, you can instantly see there are many more purple cells. So there's an expansion of lymphoid cells within the blood. And the problem there is that if your stem cells are making more lymphoid cells, they're not making enough myeloid and red blood cells. And so often one of the first symptoms of leukemia is tiredness and lethargy because you haven't got enough red blood cells to carry oxygen around your body. So that's leukemia. As I say, we're going to focus on lymphoma. Lymphoma is a solid mass. And as we know, lymphoid cells travel through the lymphatics as well as the blood, and they congregate at sites of lymphoid organs. So spleen, the thymus, which of course, you know, sits on the heart and makes the T cells, our tonsils, lymph nodes in our armpits, in our groin, up here, we have lymph nodes everywhere. So anywhere in the body, a lymphoma can develop. And as I said before, often swollen lymph nodes mean nothing. They just mean you have an infection. And within a couple of weeks, as that infection clears, the lymphoid cells will die and the swelling will go down. Now, when those swollen lymph nodes stay swollen for a longer period of time, it can be a sign of something more sinister. It can just be a lingering infection, but sometimes it can be something more serious, such as a lymphoma. And you can see in the boy on the top there, large masses in the neck. But lymphomas can also present in a cutaneous form in the skin. And you can see the lady on the bottom picture there has a cutaneous lymphoma in her cheek. So what are lymphomas? Well, they're categorized into two different subsets. We have non-Hodgkin's lymphomas and we have Hodgkin's lymphoma. And Hodgkin's lymphoma I won't talk about. That's a very distinct type of lymphoma on its own. It's associated with something called a Reed-Sternberg cell, which is that big cell in the picture in the middle there. And actually, Hodgkin's lymphoma, most of the tumor cells are reactive lymphoid cells. Sorry, most of the tumor is reactive lymphoid cells, whereas the malignant cells are just a very small percentage of that, and they're the Reed-Sternberg cells. So it's a very unusual cancer. It's very rare in the UK. It occurs more in males than females for reasons we don't understand. But it also seems to occur more often in the third decade of life, certainly in the Western world. What I'm going to focus on is non-Hodgkin's lymphoma, which in itself is at least 40 different diseases. And you'll see why it's 40 different diseases shortly. In the UK, we have about 20 cases per 100,000 of the population. So it's the sixth most common cancer in the UK. So that's about just over 8,500 cases per year. There's a male predominance, and again, we don't fully understand why that is, but it is in general a cancer of old age. It's diagnosed by a number of ways. So if there is a swollen lymph node, the first thing a doctor would do would be to take a biopsy. So to take some cells from that lump and look at them under a microscope. And the first thing they would do is look for the clonality of that population of cells. So as we know, B and T cells rearrange their antigen receptors. And if they're responding to an infection, you would expect to see polyclonality, so lots of different rearrangements of T cell and B cell receptors. Now, if it's a tumor that's expanded from one clone, you would expect to see all those cells to express the same rearrangement of the T cell or B cell receptor. So if we see monoclonality, that gives us an indication that this may be a lymphoma as opposed to an infection. What would then happen would be a histopathologist would look at the architecture of the tissue and see if it looked abnormal in appearance, would look at its immunophenotype, so are these B cells or T cells, so we could determine which type of lymphoma it is. And these days, more and more, we're looking at genetic profiling of tumor cells. So on the right, there is a microarray looking at the RNA transcripts within the tumor cells, and we can align the RNA expression profile to that of normal lymphoid cells to try and work out their cell of origin. So for example, in a type of lymphoma called diffuse large B-cell lymphoma, sometimes those tumor cells have an RNA expression profile which makes them look like activated B cells. And sometimes they more look like germinal center B cells. And so therefore we can give a diagnosis to those tumors based on their genetic profile, which could lead to different treatments. Lymphomas can be aggressive, so they can be very fast growing like diffuse large B-cell lymphoma, but some lymphomas are very indolent, so they're very slow growing. An example of this is a follicular lymphoma. And follicular lymphomas, we actually don't even treat them. We do something called watch and wait. We just monitor them. And sometimes they can do something called transform and become an aggressive diffuse large B-cell lymphoma. So those are just watched rather than the aggressive tumors, which we have to treat with chemotherapy. And as I said before, they could present at any site in your body, wherever there are lymph nodes. And sometimes the symptoms can be bone pain, rashes, weight loss, anything like that. And sometimes they even present at extranodal sites, so skin, liver, all around the body, essentially in any organ. Patients also sometimes get something called B symptoms. And if you think about what immune cells normally do, they are normally producing cytokines, chemokines, things that increase our body temperature, things that cause us to lose our appetite. And so all those symptoms normally associated with an infection, such as flu or a bacterial infection, can also be the symptoms of a lymphoma. So again, it's because we're getting expansion of lymphoid cells which are producing all those factors which lead to those symptoms. So this just repeats what I said before. They can be nodal or they can be extranodal. And so basically lymphomas can present anywhere in your body. So sometimes people may, for example, have a tumor in the breast and it may be mistaken for a breast cancer. But when a histopathologist looks at the tumor and does the immunophenotyping, they would then be able to say whether this is a lymphoma or a breast tumor. We also have different staging of the disease. So stage 1 is where only one lymph node is involved. And obviously the lower the stage, the better your prognosis. If just one lymph node is involved, it may be possible just to surgically excise that lymph node and then the cancer's gone. But if it gets through the stages, for example, 1E is where you have an extranodal site of presentation, such as the skin or the liver. That's much more difficult to treat with surgery. Stage 2 is when you have involvement of 2 lymph nodes. Again, can still be perhaps treated with surgery, but more likely is going to need chemotherapy because the implication is there that tumors have moved from one lymph node to another and therefore have started to spread. Stage 3 is when lymph nodes either side of the diaphragm, and stage 4 is the worst prognosis stage, and this is where we see involvement of lymph nodes and other organs, so spread of the tumor, and these patients for certain need chemotherapy and radiotherapy. So again, we're talking about cells on the right side of the hematopoietic tree there, the lymphoid cells. And we diagnose lymphomas based on the type of cell we think they arose from. So did it arise from a natural killer cell? Did it arise from a T cell or a B cell or a plasma cell? That would then be called a myeloma, which is again a different cancer. So we won't talk about cancers of the plasma cells. Or maybe the initiating genetic events actually happened in a common lymphoid progenitor, a cell that already has the ability to self-renew. In which case, those cells are already one stage towards developing the hallmarks of cancer because they can already self-renew. Whereas our more differentiated cells would have to acquire mutations which would give them the ability to self-renew. So basically, broadly speaking, we have B-cell lymphomas, we have T-cell lymphomas, and we have very rare NK cell lymphomas or even NK T-cell lymphomas. And the B-cell lymphomas account for the majority of lymphomas that we see. And I personally think that that's probably because B cells inherently go through a number of processes that induce DNA damage. Somatic hypermutation, class switch recombination, rearrangement of the immunoglobulin genes. All of these processes are natural ways of inducing DNA mutations, errors in the DNA code. So I think this is why we see more lymphomas in B cells than we do T cells, because in comparison, all T cells do is rearrange their T cell receptor genes. And they're further characterized, further broken down into the type of cell we think they may have arisen from. So there are a number of different lymphomas, and sometimes they're given the name they're given due to the site they're seen in. So sometimes lymphomas arise in the mucosal associated lymphoid tissue, and therefore they're called MALT lymphomas. Sometimes they arise in and grow in a follicular pattern, much like a germinal center, secondary follicle, so we call them a follicular lymphoma, and so on. So there's a range of different names for different types of lymphoma. But broadly speaking, we name them according to what stage of B cell or T cell development we think the transformation event occurred in. And this is helped in lymphoma because, of course, we know that certain CD proteins are expressed on cells at certain stages of development. So if we look at the lymphoma cells, and we can say they express CD34 and CD20, that gives us some idea about the stage at which they were when the transformation event occurred. Okay.
When you get to a point you want to have a pause, do have a break if you want.
Okay. Okay. So if we think about a normal lymph node, this is the structure of a lymph node, and there are lots of different cell types within there, but broadly it's broken down into the medulla, which is the region where we have the sinuses and the efferent lymphatic coming in, and the cortex. And within the cortex is where we have the follicles. So primary follicles, which aren't undergoing any sort of immune response, and then the secondary follicles, which are the cells where you have a germinal center, where the cells have been responding to some sort of presented antigen by follicular dendritic cells or B cells. And these cells are responding and dividing. And then you have the mantle zone around that, which are the cells which aren't responding to the antigen. So if we look at that germinal center under the microscope, and I'm just going to put some rings on there so you can clearly see the different areas. There's that germinal center in the middle. So they're the cells which are dividing, they're responding to antigen. You then have that mantle zone. Those are the cells which aren't responding to antigen, that aren't dividing, and so they're still quite nice small lymphocytes. And then you have the marginal zone around it, and then of course you'll have lots of these germinal centers. And some lymphomas are categorized based on where we think they come from. So some lymphomas look like mantle cells in that they are not activated B cells. And so we call that a mantle cell lymphoma. And the other way we characterize that type of lymphoma is they have chromosomal translocations, which juxtapose the heavy chain enhancer, so EMU, to BCL1, which is the an anti-apoptotic gene. So therefore, you have cells with a genetic event which prevents apoptosis and enables them to survive. So we call these mantle cell lymphomas. Equally, tumors that arise from that germinal center, we call follicular or Burkitt's or diffuse large B-cell lymphoma, depending on the type of chromosomal translocation they have. And you'll notice that in B-cell lymphomas, the most common translocations involve chromosome 14. And that's where the heavy chain enhancer sits. So again, you can see why these B cells may be prone to cancer development is because they're already rearranging the VDJ genes. And therefore, the heavy chain enhancer in some cases is randomly being joined to a gene which it shouldn't be. So either MYC in the case of Burkitt's or one of the BCL apoptosis proteins in the other types of lymphoma. So you get aberrant expression of these proteins which are preventing apoptosis, or in the case of MYC, are driving cell proliferation. There are some further types. So malt lymphoma involves the cell in that area around the germinal centers and around the secondary follicles. There's a CD5 positive small lymphocytic lymphoma and a PAX5 positive, again through a T14 translocation, lymphoplasmacytic lymphoma. T-cell lymphoma, on the other hand, is slightly different. So with T-cell lymphoma, we divide it into tumors that arise from immature or mature T cells. And immature T-cell lymphomas are called lymphoblastic lymphomas. They arise from the thymocytes within the thymus, and we see that very often in children. On the other hand, the mature T-cell lymphomas, the peripheral T-cell lymphomas, the anaplastic T-cell lymphomas, they arise, we think, from mature T-cells circulating in the periphery. So from your helper T-cells and your cytotoxic T-cells. Okay, so what causes lymphoma? So we know about those genetic mutations. We know about the events that happen in lymphoid cells, so the VDJ recombination, somatic hypermutation, class switch recombination. And you can imagine that if you've had a lifetime of your B cells or your T cells undergoing these processes and dividing and then stopping dividing and creating memory cells, and of course memory cells reacquire the ability to self-renew. Another process which is going to aid lymphomagenesis. And what fits with that is we know that most people that develop lymphoma develop it in later life. So this fits with a lifetime of acquiring mutations in your lymphoid cells. And much like many of the other cancers, we know that lymphomas can be caused by a number of different factors. So those mutations can be caused by an integrating retrovirus, for example, and I'll give you some examples of that shortly. Some lymphomas have been shown to occur from epidemiological studies in certain communities. So we know some lymphomas are more prominent in farming communities where, for example, organophosphates are used. We know that some lymphomas are genetically acquired. Again, it's very rare. We also know that some tumors arise in the context of a chronic infection. So there's some sort of chronic antigenic stimulus driving lymphoid cell proliferation, perhaps leading to more replication-induced DNA damage, which could be driving tumor development. We know there are chromosomal translocations in many lymphomas, so those heavy chain enhancer rearrangements. So are those causing the driving initiating event? And I say chicken or egg here because we don't know if maybe those chromosomal translocations are the consequence of genomic instability rather than the ones that are driving genomic instability. And it could just be bad luck again, inappropriate VDJ recombination, where the heavy chain, when it gets cleaved for that VDJ recombination, instead of being rearranged in the correct place, it's actually getting juxtaposed to other genes which happen to be transcribed at the same time. So for example, genes such as MYC. Okay, so lymphoma and infection. So there are lots of different types of lymphoma associated with infection, with not just viruses, but also with bacteria and also with parasites. So for example, Helicobacter pylori is known to cause a type of cancer called malt lymphoma, so mucosal associated lymphoid tissue. So much like we have a lymphoid tissue throughout our bodies, the lining of our gastrointestinal tract has lots of germinal centers. This is called MALT, and these are there, of course, as a barrier to protect our intestines, which are exposed every day to pathogens from moving further out into our bodies. We know Burkitt's lymphoma, as is very common in Uganda, is associated with EBV infection. And these top two I'm going to go into more detail about, but there are other types. So extranodal NK T-cell lymphoma in the nose is also associated with EBV infection, but we see that more in Asia. We know that patients who have an allergy to gluten and have developed celiac disease are more prone to develop lymphoma. And that's a type of lymphoma called enteropathy type T-cell lymphoma. We know that patients who have a suppressed immune system, so for example, if a patient has had a heart transplant and is taking immunosuppressive drugs to prevent rejection of that heart transplant, they are more at risk of developing lymphoma. Human T lymphotropic virus, HTLV, causes an adult T-cell leukemia lymphoma. We know HHV8 and HIV are associated with myeloma, the plasma cell lymphoma. Diffuse large B-cell lymphoma sometimes arises again in the context of immunodeficiency. And also some patients who have HIV, for example, and then develop a KSHV or HHV8 infection are at risk of something called primary effusion lymphoma or PEL. So those immunosuppression-related lymphomas, we see lots of those in patients who have HIV and therefore have a suppressed immune system. So there are no T cells essentially in these patients controlling B cell growth. So these patients are then predisposed to viruses which drive hyperproliferation of their B cells, but they have no T cells to control that. So these are predominantly aggressive B cell lymphomas, as I've previously mentioned. And it can, in some patients, be the first defining symptom of AIDS. So these HIV patients sometimes aren't diagnosed with AIDS until they develop the lymphoma. And with these, again, it's probably due to a chronic antigenic stimulus driving hyperproliferation of the B cells, which enables them to acquire further mutations and become malignant. And in many of these lymphomas, we see mutations in your common oncogenes and tumor suppressor genes, such as MYC, RAS, P53, and BCL6. And again, the viruses we think they're exposing them that are driving that chronic antigenic stimulus are proposed to be EBV, KSHV, and HHV8. In a similar manner with the post-transplant lymphoproliferative disorders, again, we have immunosuppression in these patients. And what we think happens there is that we are all infected with EBV, but normally it's a latent infection. And that latent infection becomes active when there is immunosuppression. And so we think this is the mechanism in these patients, is that EBV gets reactivated on immunosuppression and can drive chronic cell proliferation of the B cells. And it commonly occurs, if it is going to arise, within 2 years of transplantation in these patients. In celiac disease, it's slightly different. So patients who have celiac disease are very sensitive to a protein found in wheat, a gluten protein called gliadin, and they're at an increased risk of lymphoma development. And again, what we think is the problem with these patients is that their lymphoid cells are hyperactive in response to gluten, which is the antigen, and this therefore drives hyperproliferation, in this case of T cells, and they get something called enteropathy type intestinal T cell lymphoma. But the risk can be reduced if these patients stick to a gluten-free diet, if they don't provide that antigen to their T cells. So we're talking about lots of viral infections with lymphoid malignancies. So often EBV infection in immunosuppressed patients drives cancer. And this is likely due to a defective immune surveillance to that Epstein-Barr virus. We think that perhaps this latent EBV infection can be transferred to patients who have, for example, bone marrow transplants, and that can also, in those immunosuppressed patients, drive lymphoma development. But ultimately, what it comes down to is a form of chronic antigenic stimulation, which leads to a lymphoid hyperplasia, so an abnormal growth, which then acquires further mutations, giving those hallmarks to the cells, and they become these malignant lymphomas. Okay. Do we want to take a quick break?
Yes.
Do you want to take a quick break?
There might be some questions that people want to ask at this point.
Okay. We'll take a little break there before I go into the bacterial driven lymphomagenesis.
Questions now or later?
If you've got questions, I'll take questions.
Has anybody got any questions at the moment they would like to ask Dr. Turner?
Yes, there is a question. Hello. You need to be clear. Thank you for the presentation. I just wanted to know, can a glucose-free diet improve the outcome of cancer?
Did you hear it? Sorry, can you repeat it?
Could a glucose-free diet improve outcome of cancer?
A glucose-free diet? Yes, so it's interesting, isn't it? Because this has become a really active area of research in cancer at the moment, and it's the whole area of metabolomics. So how are cancer cells undergoing metabolism, and does it differ from how other cells metabolize things? And there have been studies, and I have to say I'm not an expert in this at all, but there have been studies showing that cancer cells metabolize things such as glucose in a different way to other cells. And therefore, if we starve them of the glucose, that therefore you may in some ways be able to help with the chemotherapy. So I think it's still very early days in the research about this, but I would think that it wouldn't harm patients to perhaps have a diet that was reduced in glucose, but I wouldn't use it as a substitute for any form of therapy. So certainly you would go ahead with your chemotherapy or your radiotherapy, but I don't think it would do any harm to do that. Does that answer your question?
Yeah, it does. The second question, I talked about blood supply to the cancer cells, cutting off blood supply to the cancer cells. How do you suppose that is done?
How can you cut off the blood supply to cancer cells?
Yeah, so there are lots of anti-angiogenic therapies about now. So there's one called Avastin, a drug called Avastin, which has been around a long time now. And one thing you can do is to stop vascular endothelial growth factor from binding to its receptor. So if that can be done, then you prevent the growth of those blood vessels any further. So that's the sort of thing people are looking at, is anti-angiogenic factors, such as things which prevent vascular endothelial growth factor from binding to its receptor. And those drugs are being used to treat breast cancer and have had some level of success. But again, I think with cancer, you need to take a multiple approach. And you can't just treat with one drug that just blocks angiogenesis. You also need another drug that kills the tumor cells, and maybe another drug that activates the immune cells. And so going forward, we're probably looking at combination chemotherapies.
Whoops. If we can get the slides back up rather than our emails.
They're on the screen.
Thank you, Dr. Suzanne. I also have a question. There are two questions actually. One, I'm wondering, how do cancer cells acquire immortality? Then the second question is, do the NK cells and cytotoxic T cells have a serious role in controlling cancer? If they do, then how is it that cancer is actually attacking the site for the soldiers?
Yes, so immune cells do in some cancers play quite a big role in keeping the cancers at bay. And it may be that many immune cells, we don't see some cancers because our immune system is dealing with them through NK cells and T cells. But what we know that some cancer cells do is they avoid being seen by the immune system by essentially changing expression of the PD-1, PD-L1 signaling axis. So this essentially tells the immune system that they're normal cells and not to kill them. And so there are drugs now which are being used to reactivate that pathway so that essentially the immune system can now see the tumor cells and attack them. So that's a very active area of research at the moment. It's in its very early stages, and I guess we'll wait and see how that develops. But there are certainly many groups trying to reactivate T cells and also now NK cells, although that's a much earlier stages of research than it is with T cells to do that. I can't remember the first question. Sorry, what was the first question?
The first question was, how do cancer cells acquire immortality?
Yes, so one thing cancer cells do is they increase the length of their telomeres. So some cancer cells acquire mutations in telomerase, and telomerase then increases the length of the telomeres. And as you know, as cells divide, our telomeres normally shorten. And we think that, you know, very short telomeres can trigger apoptosis. So one thing they do is to increase the length of their telomeres. And the other thing is any pathway essentially that will inhibit apoptosis and drive proliferation will give them that sort of immortality. So it's the loss of those checkpoint inhibitors such as P53 and retinoblastoma or P16. Okay.
Thank you, Dr. Suzanne.
You're welcome.
I'd like to take you back to when you talked of reactive oxygen species.
Yes.
So for these microorganisms that are associated with cancer, are they also associated with a high level of reactive oxygen species?
That's a fantastic question. And I don't think anybody has looked, to be honest, but no, that is a fantastic question. Yeah, really good question, and I can't answer it, I'm afraid. I don't know if anybody's looked to see if that's the case or not, but I wouldn't be surprised. I know that in the case of colorectal cancer, the cancers that have higher levels of reactive oxygen species are of a worse prognosis. And I know that there are people looking at using drugs to enhance reactive oxygen species as a way to kill the cancer cells. But I don't know if anybody has looked at it in the context of infections. But no, fantastic question, but I'm afraid I can't answer it.
Okay, then the second question is about increased metabolism and oxidative stress. So when you talk about metabolic stress and oxidative stress as drivers for cancer, and yet these are the actual things that happen during exercise, when you tell people to increase their physical exercises, I expect the body to similarly increase the amount of metabolism, since the metabolic stress. So
yeah, absolutely. So in that case, it's in the context of the cells being normal and healthy and being able to respond to that stress in a normal manner. In tumor cells, it's considered an enabling property to deal with it, because they have to sort of upregulate all the pathways that you would if you were doing exercise. So I don't think it's not a hallmark that's necessary for cancer cells, but it's one of those properties which we think they acquire. So I don't think it's not cancer causing in itself, but when you're an established tumor cell, it's something that can help you to survive your environment. Does that make sense? So yes. Carry on exercising. It's good for you.
Okay, and then something else about the exposure to infectious agents like the microorganisms. In countries where infectious diseases are endemic, so we'd expect the B-cell cancers to be higher, the B-cell cancers and T-cell cancers to be higher, more so in children who are being exposed at an early stage.
Yes.
So is that the reason why there is a higher prevalence of those cancers in Africa due to the increased exposure?
Yes, so we do have some EBV-driven cancers in the Western world, but as you're right, they're much more prevalent in sub-Saharan Africa. And we think that's because of the malaria and malaria driving immunosuppression. And I'm actually going to talk about this in a few slides. So maybe I'll just save the answer. And if I don't answer it in the rest of my talk, we can come back to it.
Okay, thank you.
Yeah.
There are more questions, but I think they will come at the end. We can continue.
Yes, let's carry on. So I'm going to tell a little bit about bacterial driven. Stop doing it. Oh, there we go. Bacterial driven cancer development, because I talked about this mucosal associated lymphoid tissue lymphoma, which is associated with Helicobacter pylori, which is a bacteria. And Helicobacter pylori is the bacteria that causes gastritis, so stomach ulcers. And I don't know if you know the story of the Australian scientist that discovered this by himself drinking Helicobacter pylori, and then he developed a stomach ulcer. And so this is how he proved that this bacteria caused stomach ulcers. So not a way you want to prove anything to do with cancer, I'm sure, but certainly this gave us some ideas about how bacteria might be driving lymphoma development. So what you can see here in the top left is a normal gastric mucosa, and you can see it's mostly those glands again. And then with an infection with bacteria, so the brown staining is the bacteria. That's just highlighting that there's bacterial infection in the gastric mucosa. And can you see that germinal center that's just there in the middle of it? So the pale center with the ring of lymphocytes. Oh, okay, yeah. So this germinal center here, this is mucosal associated lymphoid tissue. And you can see there's a germinal center. So it's obviously responding to some sort of antigen. And of course, what we know from this staining is that there is a bacterial infection. So therefore, that's what it's most likely responding to. And if this persists, this bacterial infection, you can develop something called a low-grade malt lymphoma. And you can see here that now we've lost that nice ordered structure where we have all those nice glands. And now we have this big mass of cells which has grown out here. And this is the cancer, this is the lymphoma. And it's arisen from these reactive germinal centers. And in time, this can become much more aggressive and transform into something called a large B-cell lymphoma, which is much more difficult to treat. At this stage, it's much easier to treat because the tumor cells are proliferating still in response to the bacterial antigens. So what is the evidence that bacteria causes cancer? Well, it was shown in northern Italy, for example, that patients who had a high level of H. pylori-induced gastritis, so infection of the gastritis, had a higher risk of malt lymphoma. And they showed this association in a case-controlled study. There were also some in vitro studies done which showed that gastric malt lymphoma cells, if you expose them to H. pylori, they expand, they divide and proliferate, which suggests that that is driving their growth. We also know that if you eradicate bacterial infection in some patients by treating them with antibiotics, you cure the lymphoma. So that suggests, again, that this bacteria is driving the proliferation and driving the lymphomogenic process. And we also know from animal models that if you infect these mice with Helicobacter pylori, they get this chronic gastritis, and in time they develop malt lymphoma. So this is sort of the cause and effect, if you like, looking at Koch's postulates, showing that you can induce a cancer by infecting with the bacterial agent, and on the other spectrum, that if you look for the bacteria in the lymphoma cells, you see it there. So what's happening within this tumor? So this is a B-cell lymphoma, and it's quite interesting because it seems these B cells are responding to T cells which are specific for Helicobacter pylori. So this is very much an interaction between B and T cells, and it's the T cells which are presenting through the T cell receptor antigen from Helicobacter pylori that are driving the B cells into proliferating. And they do this through the production of cytokines, so in particular BAFF and CD40 ligand, which also bind to the surface of the B cells there, and in doing so also activate NF-kappa B pathway, which drives cellular survival. We also know that these are B cells which are responding to autoantigen, which is conversely activating the other arm of NF-kappa B, again driving pro-survival. So what you end up with is a tumor, a malignancy that is growing due to a bacterial infection, but not a bacterial infection of the tumor cells, but the T cells are presenting that infectious agent to the B cells, driving their proliferation. So at this stage, this cancer is very much dependent on the T cell help, okay? And you can see that here. So on the left, going back to that picture of the gastrointestinal tract, so quite healthy there on the right, like I showed you before for the colon cancer. But you can see it on the left here, these rather large, nasty lumps. And this isn't a colorectal cancer, this is a lymphoma. So this is expansion of that malt tissue. And you can see in the bottom left picture there how messy and disorganized the growth is. And this patient with this rather nasty, aggressive looking tumor is treated purely with antibiotics to kill off the Helicobacter pylori. And within 10 months, on the picture on the right there, you can see the tumor's gone and everything's back to normal. So this is an example of a bacterially driven lymphoma. But we know that some of these tumors in time can become independent of the bacterial stimulus. And this happens because they acquire mutations in their DNA. And in particular, they acquire a chromosomal translocation, as we discussed before. So the swapping of the DNA, and in particular, this is due to activation of the NF-kappa B pathway, independent of outside-in ligand-derived signaling. So this has been shown quite nicely in this publication, where patients that respond to antibiotics do not have the translocation, as you could see on the right-hand side of that table there. So the patients that go into complete remission on antibiotic treatment, very few of them have this advanced chromosomal translocation, where the patients that don't respond to the antibiotic, their tumors have therefore become independent of the bacterial stimulus because they have this chromosomal translocation, and these patients have to be treated with antibiotics. So essentially, chronic Helicobacter pylori infection can give rise to acquired mucosal associated lymphoid tissue. Some patients with this chronic stimulation acquire further mutations and translocations, which gives them a transformed mold lymphoma, which is independent of bacterial infection. Some of them acquire this through the pathway of firstly being dependent on H. pylori and later becoming independent. And we think there's also a route in these arms on the right here where they just develop this translocation independent of any chronic bacterial infection. So chromosomal translocations are very important in lymphomagenesis, and they're associated with a wide array of different types of lymphoma, often different types of translocations. And so at the top there in this table, you can see that most of those translocations are associated with inappropriate expression of a protein following fusion to the heavy chain enhancer EMU. So in particular in Burkitt's lymphoma, these MYC-associated translocations. So that brings me back to viral-driven lymphomagenesis, and in particular, the case of Burkitt's lymphoma. So how do viruses drive lymphoma? There are a number of mechanisms. The most easiest to explain is the viral genome integration. So things such as HTLV, retrovirus, which can integrate into the genome. If it integrates into oncogenes or tumor suppressor genes, it can cause their activation or inactivation respectively and drive cancer growth. So that's one mechanism. Another mechanism which we've come across and we've talked about, this chronic viral infection driving proliferation, and that proliferation predisposes the development of acquiring further mutations and cancer development. But there's a third one, which is hit and run, which is where we may see no evidence of the virus in the established tumor, because by the time the tumor is developed, the virus has gone. And I just illustrate that in a sort of cartoon form. So for example, if we have a B cell that is infected with a virus and is presenting viral antigens, and a T cell that is responding to those viral antigens, but the T cell itself isn't infected, and those T cells proliferate and become a tumor, but then we don't ever see the B cells that are infected with the virus, this is the hit and run mechanism. And the bottom picture there just shows that integration of the viral genome activating oncogenes and tumor suppressor genes. So we may or may not see evidence of the virus in the established tumor. Now with Burkitt's lymphoma, endemic Burkitt's lymphoma, we do see evidence of the virus in the tumor cells. And I'm sure you're all familiar with the story of Burkitt lymphoma, as Kampala is the home of the discovery. So this was Dennis Burkitt, who was over in Kampala in 1957, and he noticed these jaw swellings in the children and tumors in the abdomen. And when you look at them under the microscope, they look like what we call small round blue cell tumors, and I'll show you a picture shortly. And he originally published this thinking it was a sarcoma, and a sarcoma is a bone cancer. So he thought these were bone cancers in the jaw. But he went on to define it in more detail, and he defined something he called the lymphoma belt of sub-Saharan Africa. And he defined this as an area that was 10 degrees north and 10 degrees south of the equator. So that area in between, around the equator, where there was a mean minimum temperature of 60 degrees Fahrenheit and a yearly rainfall of greater than 50 milliliters. And he called this the lymphoma belt. And of course, we also know that this overlaps with the malaria belt, okay? So this was the first indication that malaria might be involved, this parasite might be involved in the lymphomagenic process. And he was quite an interesting man. He even treated some children in Kampala in his kitchen by giving them a drug called methotrexate. But he really helped to define what was driving this cancer development, because he sent some of these tumors to Professor Epstein in 1961. And Professor Epstein isolated the Epstein-Barr virus, EBV, as we all know it now. And this was the first evidence of a virus being involved with a human cancer. And that was back in 1963. And this was then named Burkitt lymphoma after Dennis Burkitt, who of course first described Burkitt lymphoma. And that was published quite some time ago now, back in the 60s, 1964, 1965. So the first evidence of a virus driving what was then became a lymphoid cancer in the jaws of children who live within the malaria belt. So this is what Burkitt lymphoma looks like. And you may have come across children in Kampala or in Uganda who have endemic Burkitt's lymphoma. And you can see the swelling of the jaw there. And it is in these malaria endemic regions. We see EBV infection in these children and also activation of the protein MYC. And MYC drives proliferation. And that's the consequence of that heavy chain enhancer MYC translocation. So you get hyperactivation of MYC. There's also a form of Burkitt's lymphoma called sporadic Burkitt's, and that's what we see in the Western world. And this little girl here is called Francesca. And unfortunately, Francesca didn't survive. She had Burkitt's lymphoma. It's very aggressive. It's the fastest growing human tumor we know. It doubles every 24 hours. It's very, very aggressive. So in the Western world, we treat these children with very aggressive chemotherapy, so lots of different drugs, and very often they end up in intensive care on life support. But survival is really high. It's over 90%. Unfortunately, not for little girls like Francesca, but it's very rarely associated with EBV in the Western world, and certainly we don't have any malaria prevalence. And the picture on the center there, that's what Burkitt lymphoma looks like under the microscope. And it looks the same, whether or not it's taken from a child in Africa or a child in the UK. When you look at it under the microscope, it looks exactly the same. And that is this so-called starry sky appearance, where you can see these macrophages, which are supposed to be the stars. So you have to use your imagination a little bit. And all these small, round blue tumor cells around the macrophages. So there are lots of similarities, but also differences between sporadic and endemic Burkitt's lymphoma. Endemic Burkitt's lymphoma is much more prevalent. So worldwide, there's just under 8,000 cases a year, and most of those are associated with EBV. Sporadic Burkitt's is much rarer, so we only see 400 cases worldwide, and only 25% of those will be attributable to EBV. Survival differs as well. So in the Western world, survival is approaching 98%, whereas in most African nations, survival ranges from 10 to 70%, depending on the region. And that could be because we have access in the UK, we're very lucky to have access to intensive care facilities, which means we can treat the children very aggressively with lots of different chemotherapeutic drugs. Whereas in most African nations where you don't have access to intensive care, you cannot give the children that aggressive chemotherapy. So they often get just 1 or 2 drugs. But it could also be the case that they are slightly different diseases. And even if we gave children in Africa the very intensive chemotherapy, they may still not have the same survival. But this is what we need to do research for, to try and uncover exactly what is endemic Burkitt's lymphoma. But what we're certain of is that EBV in malaria endemic regions predisposes children to Burkitt lymphoma. So we're dealing with 2 different problems in the different populations. For endemic lymphoma in Africa, we're probably under-treating children. We need to find ways of increasing the doses of chemotherapy that we give to children, and maybe need to think about trialing new drugs. Whereas in the Western world, in the UK and in Europe, we're probably over-treating many of the children. We're probably giving them too much drug, and we need to think about ways we can de-escalate that drug. and also to think about how we can trial new agents. And so I think by studying both the endemic form and the sporadic form, we'll be able to help each other with the problems that we currently face. So what happens with Burkitt's lymphoma, with endemic Burkitt's lymphoma? What we think happens is that there is this latent EBV infection in B cells. And then when a patient is immunosuppressed, and that can either be through malaria infection or HIV infection, whereby in both cases there is a reduction in T cells, allows those B cells with an EBV infection to proliferate. So the T cells are no longer there to keep them in check. So you get expansion of the germinal center, and you get ongoing proliferation of B cells in the germinal center driven by EBV, and this predisposes the cells to developing chromosomal translocations, in particular translocations involving the heavy chain enhancer, EMU, whereby you get activation of MYC. But that's not the full story, because if you express MYC, activate MYC in any system, you don't get full transformation. So something else must be happening. And so it's also thought that some of the gene products of EBV also add to the process. So whilst we have MYC being driven and we have T cells being suppressed, at the same time, we have viral genes which are enhancing tumorigenicity by inhibiting apoptosis. And a combination of all these processes ultimately leads to the development of endemic Burkitt's lymphoma. Here's that important translocation, which we see in children with Burkitt's. So this is where you get the swapping of the genetic material. So the MYC gene, which is on chromosome 8, becomes juxtaposed next to the heavy chain enhancer, and we end up with a new gene essentially. And this new gene is the MYC oncogene, now being expressed in these cells because it's under the control of the heavy chain enhancer instead of its normal promoter, and you get hyperproliferation of the cells. Now, some of the first studies that were done in transgenic mice were to try and develop a model of Burkitt's lymphoma. And this was a group in Australia. And what they were able to do was express MYC in B cells under the heavy chain enhancer in mice. And what you can see is these mice develop lymphoma. So you can see this mass here under the fur. And if you open up the mouse, you can see the lymph node swellings. So these are all lymphomas. These are all the swellings of the lymph nodes. And so they could show that when you overexpress MYC in mice, they develop a B lymphoid malignancy, a B cell lymphoma. But it takes them 4 months. That's quite a long time, because you would think if you cause cells to proliferate out of control, that they would very quickly develop into a tumor. So this 4 months suggests that these cells had to acquire other mutations to become fully transformed growths, and that's the case. And that's what we think the EBV genes do, is they provide the further events, the hallmarks of cancer, which enable that uncontrolled growth, also by suppressing those T cells. So it's very much a multi-step process. So in Burkitt's lymphoma, we have an EBV infection, a latent infection, as we all do, but for most of us, that infection is kept as a latent infection by our T cells. But when malaria or HIV suppresses our T cell response, you get EBV coming through, driving proliferation, enabling the development of a chromosomal translocation that activates MYC. We acquire further tumor protein mutations, and therefore we get propagation of an established growth. And I'm very conscious of the time, and a lot of this next stuff is all early research stuff. I'm sorry, I'm just popping through it all. We won't have time for it today, but when I'm in Uganda in 10 days' time, I will present some of this research data from my lab. So I'll just end with this slide on the conclusion. So hopefully, what you'll take home from today's lecture is that cancer is very much a multi-step process, with the acquisition of number of mutations which confer on those cells the cancer hallmarks, so activate oncogenes, deactivate tumor suppressor genes, that viruses can also activate oncogenes or inactivate tumor suppressor genes, such as is the case for HTLV, the bacteria can indirectly induce cancer by causing cell proliferation and sometimes by a sort of more of a bystander effect, as is the case with Helicobacter pylori. And in another example, that viruses can drive lymphoproliferation in immunosuppressed hosts, again leading to cellular transformation through the acquisition of genetic mutations such as those leading to activation of MYC, in the case, for example, of EBV infection in patients that have malaria. And I'll end it there, and if you've got time, I'm happy to take any questions.
Thank you very much.
Okay.
Susan? Thank you, Dr. Susan, for the presentation.
You're welcome.
My concern is just to clarify on the aspect of where you said that the red blood cells are not usually affected by cancer, and then it is mainly the white blood cells. So I would like you to clarify more about that. And then the second concern, or where I need clarity, is about how the cancer cells progress from affecting maybe one tissue to the other, or one organ, or more than one organ. So that is mainly what I'm interested in.
Okay, I'll have to ask you to repeat the second question, but I'll just answer the first question first. So when red blood cells, when cells differentiate to become a red blood cell, they pick out the DNA. So red blood cells don't have any DNA. So therefore, there's nothing to generate a mutation in. And so these red blood cells, they live for a couple of days and then they just die and they get replaced. So because red blood cells don't have any DNA, they can't acquire any mutations and they can't develop into cancers. Does that answer that question?
Yeah, that is all about the first question. Then the second one is about seeking clarity on how cancer cells progress from infecting one organ to the other.
They go from one organ to the other.
Yeah.
Yes. So cancer cells can spread via two routes. One is they either enter into the lymphatic system. The second is they enter into the venous system, into the blood. So when these cells break off, you can imagine as the tumor gets bigger and bigger, cells start breaking off the edges, and then they can move to blood vessels and they can move to lymphatic vessels, and then they will move within the body according to where those lymphatics or blood vessels go. So as I said, in the case of colorectal cancer, because the colon is connected to the liver through the portal vein, tumor cells in the colon very often metastasize to the liver. In the case of breast cancer, there are lots of lymph nodes in the arm. So very often what happens is in breast cancer, the tumor cells break off from the breast and they move to the lymph nodes in the armpit. And so often breast cancer spreads to other sites in the body through the lymphatic system, and colorectal cancer more so through the blood, through the venous system. So it varies from cancer to cancer.
Yes.
I make because
Thank you, doctor. Just a question, at what time, at what circumstances the NK cells and T cells start working for the cancer cell? Like what happens in a cell? Because you were saying that what happens with the immune system, that sometimes the white blood cells can start working for cancer rather than against it.
Absolutely. So it depends on the tumor microenvironment, and we know so much more about the tumor microenvironment now than we did before, and how important it is. So you can imagine that tumor cells are secreting all sorts of growth factors and chemokines, and as are the white blood cells. And so when the white blood cells are recruited to the site of the tumor, they then also start secreting growth factors, and those growth factors can help the tumor cells to grow and survive. So it's more a case of when they do come into the tumor, they are proactive in tumour growth rather than inhibiting it. And part of that is also because the PD-1, PD-L1 axis is downregulated so that tumour cells essentially evade the immune system.
Okay, thank you.
How effective is immunotherapy against this kind of lymphoma?
How
effective is immunotherapy against this?
How effective is immunotherapy?
Immunotherapy has been a real game changer for the treatment of lymphoma. So, for example, I'm talking more about the sorts of immunotherapies that bind to the tumour cells rather than reactivate the immune system. So in lymphoma, one drug that came about 10 years ago now is something called rituximab. And rituximab binds to CD20 on the surface of B cells. And it initiates antibody-dependent cellular cytotoxicity. We also think it acts through other mechanisms, but rituximab essentially leads to death of the B cells, these B cell tumour cells. So we call that a form of immunotherapy as well. There's another version which acts on CD30 on T cells. That's called brentuximab. But with brentuximab, the anti-CD30 antibody is loaded up with a mitotic spindle poison. And when it attaches to the tumour cells, because they express CD30, it gets internalized. And then the mitotic spindle poison stops cell cycle and proliferation, and that kills the tumour cells. So those forms of immunotherapy have really been game-changing in the treatment of many lymphomas and have led to huge increases in survival. And I think it would be really great if we could get rituximab for use in Kampala for the children with Burkitt's lymphoma, because I think it could make a huge difference to their survival rates without causing any of the nasty side effects of standard chemotherapy. So it's really been a great asset for the treatment of lymphoma.
I think we've got time for one last question.
Okay, thank you, Dr. Susanna.
Hello.
I have a question on bacteria getting involved in causing cancer. I've been reading about on a lot of papers suggesting that normal flora may have an impact in preventing cancer, especially in the GIT cancer. I would like to know, those people who are being associated with colon cancer, is there a possibility that they may be having a change in their microflora, in the normal flora in their colon? Or do you have anything to do with that?
Yeah, the microbiome is a huge area of research now, and it's a fascinating area. It's not one that I'm involved in myself, but yeah, there have been studies that have shown that the microflora in the intestines can protect against colorectal cancer, and that maybe some people who have maladjustments in their microflora are more predisposed. It's really early stages in that area of research, but I think there is definitely something to it. And actually, what I'll tell you a really interesting story. So children with leukemia, we know that children who go to daycare when they're babies have a much lower risk of leukemia. And we think that's because of the whole hygiene hypothesis. So the children that go to nursery school and are playing with other children are being exposed to different infections. So their immune system is building up an immunity. Whereas the children that are kept at home in a very clean environment, their immune systems aren't able to do that. And so they're more predisposed to their immune systems essentially growing out of control and developing into cancers. And there's actually a guy here in London called Mel Greaves who was just awarded a knighthood actually by the Queen, but he made this discovery, and what he's trying to do now is develop excuse me, sort of probiotic drinks for children so that their microbiome is more varied and more diverse. But first we need to find out which bacteria are the protective ones, and then we'll be able to do that sort of thing. But yeah, I absolutely think there's something in that, and that studies of the microbiome in the future will really tell us a lot about what we might be able to do to prevent some cancers.
Thank you very much.